Hello, everyone. For the next 5 minutes, I'm going to tell you a little bit about how artificial intelligence and machine learning can accelerate diagnosis, drug discovery, and precision medicine with particular relevance to Alzheimer's disease. And this is joint work with many colleagues in our AI4AD Consortium on ultra-scale machine learning. So AI can be used for precision medicine. In other words, to use all the aggregated biomarker data from patients, whether it be the genetic code, omics data, neuroimaging data, or even mobility data, to make predictions about their diagnosis, their prognosis, their potential treatment response. And the way that this is done is that this data is fed into neural network algorithms, for example, deep learning algorithms, and these can be trained and used for diagnosis and subtyping in order to discover some of the features that might be most relevant for these particular tasks. Deep learning methods include convolutional neural networks, generative adversarial networks, often used with images, recurrent neural networks, often used with genome sequences. And as you can see on the top left, they often read in a certain image, in this case of a face, and they're asked to decide who it is. In this case, it's a person called Sarah, by distilling successively more abstract features, low-level features that are then built into high-level features that then eventually help the neural network decide who it is. In the medical context, we read in MRI in some cases, other types of neuroimaging data in other cases. And you can build and train neural networks that will tell you the person's age, whether they have a diagnosis of AD, or whether they might have other pathologies, coexisting pathologies, for example, those in the ATNV framework. And whether or not they're amyloid positive, for example, is very relevant, for example, if you're designing a trial of an anti-amyloid agent. And we may also need to know about other pathology. Now, AI is sometimes thought of as a bit of a black box, but you can incorporate units in these deep neural networks, such as attention mechanisms, that will produce visualizations of the features that are being used. So in this case, these are the features that are being used to decide a person's age or whether they have a diagnosis of Alzheimer's disease based on their scan. And it gives you a little bit more interpretability as to what is going on. Now, Dugu Tosun at UCSF has developed deep learning networks to identify pathology, neuropathology that normally would only be accessible post-mortem, based on in vivo markers, not just the age, sex, and demographics of the person, but also their structural MRI, different amyloid and tau-sensitive PET scans. And she's been able to do disease subtyping that with 86 to 89% accuracy identifies TDP43, Lewy body dementia, and CAA, here based on the ROS-map dataset, which both of these types of data were available. But you can imagine the value of this approach in identifying pathology and subtypes in additional people. We've also been very much involved in discovery of markers in the genome that affect brain structure and disease risk. We've discovered over 500 genomic loci that affect brain structure and over 10 that affect the speed of brain aging. But we're very interested in what are the causal loci in whole genome sequences. Are these druggable targets? What brain systems do they affect? And work by Li Shen in our AI4AD project and Gyunga Chun, who's an expert in drug repurposing, they're trying to find combinations of markers in the genomes and also combinations of systems in brain images that are maximally associated, screening all of this data to prioritize genetic markers that are druggable, lie in druggable pathways, and are easier to understand based on a whole host of bioinformatics data that is being assembled in Gyunga Chun's PSBO system. To do deep learning on whole genome sequences, a lot of data reduction is necessary. Sarah and Sasha Zaranek at Curie Corporation and Harvard University are splitting up the genome into tiles and then allowing us to feed in these methods here to Kang Huang's InterpretNet algorithm that makes inferences about disease status and prognosis based on the whole vast array of tiles coming from the genome. Some major successes of AI and deep learning are that it's now starting to be used for diagnosis, prognosis, and subtyping of pathology. You can also, as we saw in some data, identify comorbid pathologies even if post-mortem data is not available. And there's huge benefits, obviously, for drug trial stratification and identifying treatment responders using these methods. We do need to make sure that these methods work well across diverse populations. And there's a lot of enthusiasm that deep learning and whole genome sequencing will help us to identify novel drug targets based on all the aggregated work on drug action and gene action that is being assembled in bioinformatics databases. So again, thank you very much for the chance to present today.